Let's get into blood grouping and its related topic of blood transfusion. Now, when we talk about blood grouping, we must understand that the reason we focus on blood grouping is because if we don't know about it, we can't transfuse blood. So blood transfusion becomes a very big issue. So imagine a person who's injured, who's lost a lot of blood. He's lost a fair amount of blood, maybe 1.5 liters of blood. These people will go into shock very, very soon. So they will fall into low blood pressure, and they will eventually have problems with their organs functioning, and eventually they go through multi-organ failure and death in shock, because there's not enough blood to carry oxygen to meet the metabolic demands of the tissue. So one obvious answer to this problem would be, why don't we just give blood to somebody? And people tried that, but it didn't work, because people had different antigens on their blood cells. So what's an antigen first? So an antigen is a molecule that would elicit an immune response. That means that if you gave this to somebody who has not been exposed to this antigen, then there's a very high chance that his immune system is going to react and destroy or try to contain this antigen through various methods like neutralization, opsonization, which we'll talk about in immunity. Now look, all of our cells have different antigens, meaning every molecule we have in our body is built for us. Our immune system was exposed to it from our embryogenesis, when we were developing in our mother's womb. So it's not a problem. Most of the antigens are exposed to the immune system is exposed to them in our body while we are developing. And what happens is, if you give a part of your body, your tissue, to someone else, then they might not have the same proteins as you have, unless they're your identical twin, because the chromosomes and the DNA are different, so the transcription and translation would yield different proteins. So what happens is, you have these antigens which are unique to you, and if you give it to someone else, there is a high chance that their body would reject it and actually destroy it, which is not good. So that can cause hemolysis or destruction of red cells. So we must understand that our red cells have these different antigens, and they've been grouped into various different systems. Okay, so there's something called an ABO system and an RH system. This is based on the antigens which are present in it. So the ABO system has got the A antigen and the B antigen over there, and also the H antigen. So they have the A, the B, and the H antigen. I'll talk to you about this. The RH system has got 3 antigens, C, D, and E, but D is the most important of this, which we'll be focusing on. C and D, you can just forget about it. So what happens is, there are many other systems, but these two, the ABO and the RH, are the major blood group systems. They're the major blood group systems over here, and that means they're the most important when it comes to blood transfusion. If a person has the right blood group, if it matches, we can then donate blood to the other person over there. So that's why we need to know about these blood group systems. So what are these? Now, for example, we have the ABO system over here. ABO system. So this was discovered by Carl Landsteiner. When he discovered this, he was an Austrian in the early 1900s. He discovered this. We got the basis of why some people's blood transfusions are successful and some people's aren't. So he found out that on the red cells there are certain antigens which are present over there. And what happens is, if there is a particular antigen on a blood cell, which he called an agglutinogen. This was his. These are his laws, Landsteiner's laws. Landsteiner's laws. He said that if an agglutinogen is present on the surface of an RBC or erythrocyte, then meaning the antibody which would react to that antigen, its corresponding agglutinin will be absent in the plasma. That means you have an antigen. If you have an antigen on the red cell, then you can't have the antibody that would react with that cell and cause damage to that cell in the plasma, in the blood plasma. It's as simple as that. This is the first postulate, he said. Now, this is followed by all blood groups, the major blood groups and the minor ones, which I'll tell you about. There are some minor blood groups and various implications of their blood grouping systems. And then after that, the other postulate is, which again this is only followed by ABO system always. Second postulate is not always followed by other blood groups other than ABO. So I'll just tell you about it. If an agglutinogen is absent on the RBC surface, on the surface of the RBC, then its corresponding agglutinin, meaning the antibody, will be present in the serum or plasma. Same thing. Okay, this is again followed by all your blood group systems over here. By ABO system, mainly ABO system, and not necessarily by other systems. What does that mean? So imagine in the RH system, I told you we have this antigen called D antigen. If a person is RH negative, if a person is RH negative, that means by the second postulate he should have no antigen, no RH antigen, no D antigen, but he should have the antibodies against D, meaning the antibodies or the agglutinins against D. But if a person who is RH negative is born and he was never exposed to the RH antigen, he will not have antibodies necessarily in his blood. However, if he gets exposed to the antigen in life, like somebody transfuses a little blood to him which has the RH antigen, then his immune system realizes, hey, there is something foreign and I better make these antibodies. Then his antibodies are produced that time. So the RH system and most of your minor systems do not necessarily follow this rule until they are exposed to the antigen. Okay, so this is the second postulate, which is followed mostly by the ABO system only. ABO follows both postulates, but the other systems are very picky and choosy whether they follow the second postulate. There are specific circumstances when it would express itself. So your major blood group systems, ABO and RH, have different antigens. So it's based on what antigens you have in the body. If you have the A antigen, you belong to A blood group. If you have the B antigen, you have the B blood group. And if you don't have A or B antigen, you have O blood group. And you can have both antigens expressed at the same time. This is called codominant expression. So the inheritance of your blood groups, for example, when we speak about the ABO system, as an example, we find that you inherit your blood group from your parents over here. Okay, so there is codominant expression codominant expression of these different alleles over here. So for example, you might be homozygous or heterozygous, it depends, for different for an antigen. Let's say that you are the father, okay, paternal, and this is the maternal, okay, father and mother. Let's make it F and M over here, father, mother. So you have two chromosomes. Now I want you to know that the A system and the B system is dominant over O. O is recessive, unless it is like I mean, it's your A blood group. If you just have one chromosome showing A, you have the B blood group expressed if you have one chromosome with B. So let's say that this father is A blood group, so he is A, so he has the A antigen, and let's say that he is homozygous for A. So I'm drawing the A allele over here as black. Okay, so he is A. The mother can be, let's say, B antigen. So if you have the B antigen, let's just draw it in blue, green over here. So she is B, and the other chromosome can be O. Let's just say it's O. It's got the O antigen. So she is a B antigen. She has B blood group because the B is shown. The O doesn't matter. It's if the B is expressed, and that makes it B antigen. So what happens is the A and the B can combine, giving you AB blood group. The baby can be AB. The baby can be AO, meaning he is A blood group over here. AOO, meaning he is A blood group. Or he can be AB again, or he can be AO. So there is a 50-50 chance that he is AB or just plain A blood group because of this mother having the heterozygous presentation of a B blood group. If they were homozygous, again, it will change over there. So you can do the math and see over here. If another example, a father is A, but he is heterozygous, so he is AO, okay, and the mother is BO. Now you have a lot of options over here. You get AB blood group. There's a 25% chance of the child being AB, child being A blood group but heterozygous. So this is A blood group. He can have O and B, so you get the BO. So this is B blood group, and you get the OO, so you can be O blood group. So there are so many options when you are heterozygous in that way. Okay. Some people who are homozygous, however, like A for the father, B for the mother. Now these kids are all going to be ABs. No matter how you mix it, it's always going to be AB. Okay, because they're homozygous. So you see, this is how the Mendelian genetics takes place over here, and they're codominantly expressed. So A and B, if they're both present in the chromosomes, you will have A and B present over there in expressed over the A and B antigen. So they are AB blood group over there. Now, the other concept you should know, let's just make a table over here showing the ABO system and the RES system over here. So the ABO system, there are different blood groups, AB, and O. So in the ABO system, you have different antigens. The antigens present in A are, you have the A antigen. In B, you have the B antigen. In AB, you have A and B. And in O, you have a special antigen called the H antigen. So what's the story? What happens is, if this is your red cell, and you have O blood group, you have a special antigen called the H antigen. Now the H antigen does not particularly have any antibodies against it. I just want you to know this. The H antigen has no antibodies against it, really. Not particularly, unless we're talking about Bombay blood group. Just bear with me. So there's a H antigen, okay? And consider this, I like to think of the H antigen like a head, an empty bald head over there. So you have this head, and if the head is exposed, like nobody's wearing a hat, there's no hat on the head, this is the O blood group, okay? But if you put a little sugar on top of this, a little sugar, carbohydrate moiety on top over here, like for example, if you put what do we say, acetyl galactosamine, N-acetyl galactosamine, sorry about that, Galactosamine, N-acetyl galactosamine over here. This is going to give you an A antigen. Now what you have is an A antigen. By adding this little hat on top of the H antigen, you've got the A antigen. However, if you put a different kind of hat on top, this is just your galactose, the sugar galactose from milk. You get the B antigen. This is B antigen. So you see, you end up with the B antigen. So H alone makes an O blood group. When you add galactose on top of the H antigen, you get the B antigen. And when you add N-acetyl galactosamine, you get the A antigen. So this is the real story of the antigens over here, how they are made. Small carbohydrate moiety added on top over there will change the entire blood group over there, okay, based on your genetics. So now the antibody, so the antigen is over here, also known as agglutinogen, which I mentioned over here. And then you have the antibody, also known as agglutinin. Agglutinin. So the agglutinin is, if you have the A blood group, you're going to have antibodies against B antigens. So if you gave this person B blood group or AB blood group, he's going to have a reaction, hemolytic reaction, a transfusion reaction. If you have A B antigen, you are going to have antibodies against A, so you can't give them any blood which has the alphabet A labeled on it. If you have A and B antigens, you are aware, you are tolerant to both of these antigens, so you don't have any antibodies against A or B system. You can take blood from the O blood group, the A, the B, the AB, no problem. So you are a universal recipient, okay, universal recipient. Okay, so this is anti-B, anti-A, and if you are O blood group, you've not got any of these antigens A or B, so you have the antibody against both A and B, both of them. So you cannot take blood from anybody with an A or B blood group. You can only take it from the O blood group, but you can donate to all of these other people, A, B, B, and A, because you don't have you don't have any antigen which is foreign to them. They all have the H antigen. There's no antibodies against that, so that makes you a universal donor, D-O-N-O-R, okay, donor. There we go. Our next topic, very important, blood grouping. So we need to know this because we all have our blood groups, and I think most of us know our blood groups. But what is it? What are the implications of it clinically? Why can't we just donate blood to anyone we want to? Why do we have to check the blood group and match it? So this is what we'll be discussing over here. So blood groups were described by a person called Kell Landsteiner. Kell Landsteiner in the early 1900s. I think he was an Austrian. So what he found was our blood. Some people would lose blood for whatever reason, in a surgery or in battle, and you couldn't just donate blood from one person to another person just like that. There was a high chance that when you did that, the blood would start to clump and get destroyed. The blood which was donated would start to get destroyed in the person who was receiving that blood in his body. So it would actually cause more harm than good. So eventually Kell Landsteiner found out that there were some systems. The blood had some systems. There were different types of blood in a person. So our red cells had some special molecules on top of them, which are called antigens. Antigens. This has Kell Landsteiner used the word agglutinogen to describe it. Agglutinogen. Okay, we'll be talking about it soon. And there are these antibodies in the system. Sometimes antibodies will stick to antigens, causing destruction and clumping of the blood. So the antibodies are called agglutinins. U-T-I-N-I-N-S. Agglutinins. Okay, you should know this. Agglutinogens are antigens and agglutinins are antibodies. All right, what is an antigen first? Let's talk about this. An antigen is a molecule that elicits an immunological response. Immunological response. Okay? Now we all have antigens. Our immune system is used to our body's antigens. They were exposed to the antigens since we were conceived in utero itself. So they recognize our own cells as self. But if you take a tissue or transplant a tissue from another person without checking if they are similar with the profile of the proteins and the antigens, it can cause a rejection. Something like that is happening over here, and that could be called mismatched blood transfusion. So we have these blood group systems. Blood grouping is based upon blood group systems. There are different antigens or different families of antigens belonging on your red cells. And if you have a certain antigen, you have that blood group. Okay, so for example, there is an ABO system of blood grouping, ABO. So some people have the A antigen on their blood. A antigen, let's draw it in black for A. Black is A, okay? A group, A antigen. Some people have B antigen. B antigen we will draw in green over here. These are the B antigens. So people with the B antigen belong to B blood group. Okay, and there is another group of people who have got both A and B equally expressed on their cell surface.